Dear student, today's topic is histology of pituitary gland. Pituitary gland lies in a bony cavity in the sphenoid bone called sella turcica. The dimensions of pituitary gland is 15 mm transversely, 10 mm anterior-posteriorly, 6 mm vertically, and weight is about 0.5 gram. In this picture you can see the base of the skull and the location of the pituitary gland. The pituitary gland shown here with red marked circle. This is another picture of the cranial base with showing the hypophyseal fossa. This is the sagittal section of the head and neck area showing the pituitary gland. This is the picture showing the pituitary gland. Pituitary gland is suspended to the base of brain by a stalk, the infundibulum. Pituitary gland is regarded as the master endocrine gland because it regulates the secretory activity of other endocrine glands. However, the term is misleading because it is now known that the hypophyseal itself is under the control of the hypothalamus. Pituitary gland is divided into two lobes. These are adenohypophysis and neurohypophysis. Adenohypophysis, that is the anterior part, and neurohypophysis, that is the posterior part. Adenohypophysis produces and secretes most of the hormones, whereas neurohypophysis stores and releases neurohormones received from the hypothalamus. This is the pituitary gland. Two parts are there, that is the anterior pituitary and the posterior pituitary. Developmentally, pituitary having dual development origin, that is partly from the oral ectoderm and partly from neuroectoderm. Adenohypophysis is developed from oral ectoderm as an outpouching, that is called the Rathke's pouch, from the roof of stomodium or primitive oral cavity. Whereas neurohypophysis develops from neuroectoderm and it develops as a downgrowth of diencephalon. In the picture you can see the red color outpouching, that is the Rathke's pouch, and the blue color downgrowth, that is the primitive neurohypophysis. Later on, this Rathke's pouch and neurohypophysis, they join and form the anterior and posterior lobe of pituitary gland respectively. Pituitary gland is divided into two parts as already discussed, that is adenohypophysis and neurohypophysis. Now adenohypophysis having three parts, that is the pars distalis or pars anterior, second part is pars tuberalis, and third part is pars intermedia. The neurohypophysis again having the three parts, that is the pars nervosa, infundibular stalk and the median eminence. In this picture you can see the anterior pituitary having three parts, that is the pars tuberalis, pars anterior and pars intermedia, whereas the posterior pituitary having median eminence, infundibular stalk or stem and the pars nervosa. The pars anterior or pars distalis is separated from pars intermedia by a cleft or cystic cavities, which represent the residual lumen of Rathke's pouch. You can see between the pars anterior and pars intermedia in the picture there is a empty space, small empty space, that is the intraglandular cleft, and that cleft is nothing but the residual lumen of the Rathke's pouch from where the anterior pituitary gland develops. Coming to the cells of adenohypophysis, adenohypophysis consists of two types of cells, that is the chromophils and chromophobes. Chromophils contain secretory granules and stain darkly, whereas chromophobe cells contain few or no granules and stain poorly. Chromophils consists of two types of cells, that is acidophils or alpha cells, and another type is basophils or beta cells. On hematoxylin and eosin staining, the acidophil stains pink, whereas basophil stains blue. In this picture you can see in the anterior pituitary, the basophils, they are blue in color, whereas acidophil cells are pink in color. Coming to the types of chromophils and hormones secreted by them. The chromophils, that is the acidophilic cells, are two types, somatotrophs and the mammotrophs. Somatotrophs secretes growth hormone, whereas mammotrophs secrete prolactin hormone. On the other hand, basophilic cells, that is the thyrotrophs, secretes thyroid stimulating hormone, gonadotroph secretes follicle stimulating hormone and luteinizing hormone, and lastly corticotroph secretes adrenocorticotropic hormones. Coming to the functions of hormones by chromophils. First, the acidophils, acidophilic cells, that is the somatotrophs, which secrete growth hormone. Growth hormone in turn controls body growth, especially before puberty, whereas mammotrophs, which secrete prolactin, cause or control the growth of female mammary gland in pregnancy and lactation. The basophilic cells, that is the corticotrophs, secrete adrenocorticotropic hormone, which stimulates secretion of hormones from the adrenal cortex. Thyrotrophs, which secrete thyroid stimulating hormone, control the activity of the thyroid gland. Gonadotrophs, which secrete follicle stimulating hormone, cause or stimulate growth of ovarian follicles, and luteinizing hormone, which cause ovulation and development of the corpus luteum. It also causes secretion of testosterone by Leydig cells. Pars intermedia is poorly developed in human and made of colloid filled follicles, represent remnants of Rethke spout. Function of these cells in human is not clearly known. It is believed that they produce melanocyte stimulating hormone and adrenocorticotropic hormone. You can see the picture in which the pars intermedia and these pars intermedia are colloid filled. The pars intermedia, well developed in lower vertebrates, produces melanocyte stimulating hormone which acts on melanocyte cells and which in turn secrete melanin, and melanin cause skin pigmentation. Coming to the posterior pituitary or neurohypophysis. Posterior pituitary is composed of unmyelinated axons and supporting cells, that is the pituocytes. The cell bodies of these axons are present in supraoptic and paraventricular nuclei of the hypothalamus, and this causes synthesis of oxytocin and vasopressin, that is the antidiuretic hormone. The axons of neurohypophysis form the hypothalamo-hypophyseal tract, which begins in supraoptic and paraventricular nuclei. The nerve fibers present knob-like terminals. These are called herring bodies. In this picture, you can see the neurons from the supraoptic and paraventricular nuclei. In this picture shown as yellow color, and these axons form the hypothalamo-hypophyseal tract, and the ending of this tract there is knob-like structure, that is the herring bodies in the posterior pituitary. Oxytocin and vasopressin from hypothalamus transported along hypothalamo-hypophyseal tract and stored in the herring bodies. These oxytocin and vasopressin, when required, are released into the fenestrated capillaries. Coming to the function of antidiuretic hormone. Antidiuretic hormone, that is the ADH, retains body water and increases blood pressure. ADH increases water absorption in distal convoluted tubule and collecting tubule of kidney, causes decrease in urine output. It also reduces loss of water by sweat gland. ADH causes constriction of smooth muscle in arteriole, which in turn there is rise in blood pressure. On the other hand, oxytocin causes contraction of uterine smooth muscle during parturition, that is during the delivery of fetus. Oxytocin also causes contraction of myoepithelial cells of the secretory alveoli of the lactating mammary gland, which stimulates the milk ejection. Hypothalamus secretes certain releasing and inhibiting hormones or factors which control the release and inhibition of hormones from pars distalis. These releasing and inhibiting factors are the releasing factor, that is the thyrotropin releasing hormone, luteinizing releasing hormone, corticotropin releasing hormone, growth releasing hormone, and prolactin releasing hormone. Whereas inhibiting factors are growth inhibiting hormone, prolactin inhibiting hormone. Now, how these releasing and inhibiting hormone reach up to the pars distalis. This releasing and inhibiting hormone through the portal system of blood capillaries called hypothalamo-hypophyseal portal system reach to the pars distalis. The portal system, the system that carries blood between two capillary networks. Coming to hypothalamo-hypophyseal portal system. The pituitary is supplied by two sets of hypophyseal arteries, that is the superior and inferior hypophyseal arteries, and these are branches of the internal carotid artery. Superior hypophyseal artery break up into capillary network in median eminence and infundibulum and form the primary capillary plexus. In the picture, you can see the superior hypophyseal artery forming the primary capillary plexus. Long hypophyseal portal veins arise from the primary plexus, break up into secondary capillary plexus in relation to pars distalis. This vascular arrangement is thus referred to as hypothalamo-hypophyseal portal system. Now, what is the significance of this hypothalamo-hypophyseal portal system? The regulatory hormones, that is the releasing and inhibiting hormones produced by the hypothalamus via hypothalamo-hypophyseal portal system reach to the adenohypophysis and regulate the secretory activities. Thus, this is the picture showing the hypothalamo-hypophyseal portal system. Coming to clinical correlation, tumors of pituitary are usually benign and cause excessive production of hormone. For example, excess production of adrenocorticotropic hormone causes adrenal cortex to produce large quantities of corticosteroids. As a result, there will be Cushing syndrome. Excessive production of growth hormone causes in children the condition called gigantism, and in adults that is the acromegaly. Now, another condition where there is deficiency of antidiuretic hormone due to lesion of the hypothalamus, due to insufficient ADH, there will be loss of large volume of water in urine, that is called the polyuria, and there is associated with excessive thirst and compensatory water intake, and compensatory large amount of water intake is called the polydipsia, and thus this condition is known as diabetes insipidus. Thank you.